बच्चों, अब हम अपने नेक्स्ट टॉपिक पे आते हैं और वो टॉपिक है सेल और प्लाज्मा मेम्ब्रेन। इसे प्लाज्मा लेमा भी कहा जाता है। So cell membrane, plasma membrane and plasma कहाँ प्रेजेंट है? ये आउटर कवरिंग है, all the cells. When I'm saying all the cells, that means it is present in each of the cells. Every living cell has a plasma membrane encapsulating the protoplasm. It is flexible, which is very important for its functioning. It is flexible and is made up of two very important substances, proteins and a double layer of lipids. बच्चों, इन लिपिड्स को फॉस्फोलिपिड्स भी बोलते हैं, रखिएगा। These are also known as phospholipids. There were two scientists, namely Singer and Nicholson. Singer and Nicholson explained in their fluid mosaic model of cell membrane. What is the thickness? In very simple terms, it is less than 1 upon 10,000 of a millimeter. It's very difficult to imagine that thin structure. थोड़ा सा स्ट्रक्चर भी हम देख लेते हैं बेटा इसका। So this is the protein part. So same way this continues. See the protein part and lipid bilayer. क्या इन दोनों सब्सटेंसेस के अलावा भी कुछ होता है प्लाज्मा मेम्ब्रेन में? यस, इनके अलावा प्लाज्मा मेम्ब्रेन में ग्लाइको प्रोटीन और ग्लाइको लिपिड्स होते हैं। जरूर ध्यान रखना है हमें, ग्लाइकोप्रोटीन्स और ग्लाइकोलिपिड्स भी प्रेजेंट होते हैं। और देखिए, सारी बात ये है कि ये एक बाउंड्री की तरह काम है। ठीक है, ये एक आउटर बाउंड्री है सेल की, जो प्लाज्मा मेम्ब्रेन को डिपेक्ट कर रही है। मॉलिक्यूल्स का अंदर आना और मॉलिक्यूल्स का बाहर जाना, दिस इज वेरी इंपॉर्टेंट फॉर द सर्वाइवल ऑफ सेल। ये हम समझते हैं, जिस तरीके से हम अपने घर में सामान लाते हैं, जो ज़रूरत का सामान है वो बाहर से परचेज करते हैं या लाते हैं, और घर में जो भी वेस्ट प्रोडक्ट है वो हम उसे बाहर फेंक। जस्ट ऑपोजिट टू इट, टेक द एग्जांपल ऑफ अ फैक्ट्री जिसमें रॉ मटेरियल अंदर जाता है और एक फाइनल प्रोडक्ट बन के बाहर आता है। तो इस एनालॉजी से हमें समझ में आ सकता है कि प्लाज्मा मेम्ब्रेन ट्रांसपोर्ट में किस तरह हेल्प होती है। देखिए, यहाँ पे कुछ टर्म्स आती हैं, समझने की कोशिश करते हैं। इसको हम एंडोसाइटोसिस से समझने की कोशिश। Now, it is taking in bulk. This is important. Of substances. कहाँ से? From external environment. Into, taking into the cell. If solid substance is taken inside, we'll call it as phagocytosis. And if it is fluid or liquid, you can say, then it is known as penocytosis. Phagocytosis is known as cell eating. So penocytosis shall be called as cell drinking. तो बच्चों, हमने देखा सेल ईटिंग और सेल ड्रिंकिंग is the movement of solid and liquid substances into the cell through the plasma membrane. Now we'll see the next important topic, which is the transport across the cell membrane. We'll study the mechanism in the details, right? The first in this subheading is diffusion. देखिए बच्चों, डिफ्यूजन क्या होता है? कोई भी सब्सटेंस है, उसके मॉलिक्यूल्स की मूवमेंट होती है, और वो मूवमेंट होती है एक पर्टिकुलर डायरेक्शन में. अब वो डायरेक्शन क्या है? डायरेक्शन ये है बच्चों कि जहां पे उस पर्टिकुलर सब्सटेंस के मॉलिक्यूल्स की कॉन्सेंट्रेशन ज़्यादा है, वहां से वो उस एरिया या रीजन में मूव करते हैं जहां पे उनकी कॉन्सेंट्रेशन कंपैरेटिवली कम है. सो डिफ्यूजन can be defined as the movement of substances, maybe solid, liquid or gas, from their higher concentration to their lower concentration. अब क्योंकि हम बायोलॉजी में पढ़ रहे हैं बेटा, तो थोड़ा सा हमें लिविंग सिस्टम्स में डिफ्यूजन को समझना पड़ेगा. जूनियर क्लासेस में आपने एक्सचेंज ऑफ गैसेस, स्पेशली द रेस्पिरेटरी गैसेस पढ़ा है. दो कौन सी इंपॉर्टेंट गैस हैं मुझे बताने की जरूरत नहीं है, हमें पता है, दे आर ऑक्सीजन एंड कार्बन डाइऑक्साइड. ड्यूरिंग रेस्पिरेशन द ऑक्सीजन गैस मूव्स फ्रॉम आउटसाइड towards the lungs. As lungs have low oxygen concentration, similarly, the carbon dioxide moves or flows from lungs to outside, as it has a higher concentration in lungs than outside. So clearly you can see both the gases are moving from their respective regions of higher concentration to respective regions of lower concentration. After diffusion, let's now come to the next topic, which is osmosis. Now, to understand osmosis, we must understand, we should know that water obeys the laws of diffusion. Water obeys the laws of diffusion. That means some kind of movement can also occur from region where water is in its higher concentration to a region where the water is in low concentration. So how to define osmosis? I'll begin with it. It is a process in which water molecules moves from its higher concentration to lower concentration, just as it happens in diffusion, but now an additional aspect is associated with it. Through a semi permeable membrane. Now, what is it? The specifics are only solvent. We know that water is a universal solvent. Can pass from it. तो देखिए एक छोटा सा एग्जांपल लेके हम समझ सकते हैं. Let's say these are water molecules. You can clearly see the right side is having less number of water molecules, and on the left side you can see a large number of water molecules. And let's suppose this is this selectively permeable or semi-permeable membrane. What osmosis is? The movement of water molecules can be seen across this membrane in this direction, from its higher concentration to the region of lower concentration. I hope this is clear. Just to summarize, we can say that osmosis is the movement of solvent molecules. We are talking about water through a semi-permeable membrane from a region of higher concentration to a region of lower concentration. Right? Okay. So osmosis can be studied further based on the reference of direction as endosmosis and exosmosis. Do you find any similarity between endosmosis and exosmosis? The answer is yes. In both the cases, the water molecules are moving through a semi-permeable membrane from a region of higher concentration to lower concentration. Now our reference is fixed. Let's say this is my cell. You can see the water molecules being higher, or in the outside environment, you can see a few water molecules inside. Right? So clearly you can see in the external environment, the water is in higher concentration, so it will move inside the cell through the plasma membrane. So simply, what is endosmosis? Endosmosis is the osmotic entry of water into a cell or a system. So, osmotic entry of water into cell or a system. So what will exosmosis be? Let me take this as cell, which is having its share of water molecules and is placed in a region, outside region, where water deficient conditions are observed. So now you can easily tell the direction of movement of water molecules. It will be from inside to outside. So it is the exit. So I can say osmotic exit of water from the cell. Let me take water in a bowl and I put some raisins in it. And they are soaked in water. After a few hours, what do we observe? We will see that these raisins have increased in their volume upon the entry of water inside them. So this swells up. Swelled raisins. Now, can you explain the reason behind the swelling of raisins? Yes, of course. We can take raisins and the water as respectively inside and outside of a system. The water inside the raisins is in low concentration as compared to the outside condition. So, what will be the direction of osmotic movement? The direction of osmotic movement of water will be from outside into the raisin. This is a gradual process. So the accumulation of water, the imbibition of water will cause swelling in the raisin.